I think Bruno looks just the same as before. Yeah, this will be a fast talk, and unfortunately not about vision. It'll lightly touch on it. It's mostly about the hippocampus and entorhinal cortex, things that Bruno has introduced as well, and how the neural circuits in these regions of the brain allow it to perform the functions that we celebrate, like episodic memory, particularly for this talk, episodic memory. So we're going to zoom into the hippocampus and take a look at its architecture. And so CA3 is the region of the hippocampus that has this abundant recurrent circuitry that make people think that it functions as this auto-associative attractor network that stores memories. Now, where does the sensory information to be stored as memories come from? They come from entorhinal cortex, which has this nice projection directly to CA3. But before stopping at CA3, these axons first land in the dentate gyrus, and there's a second projection that also heads towards CA3. So the question from this anatomy here is that why does CA3 receive the same information from these two different pathways? All the information is coming from the same origin here, the entorhinal cortex. And there's other people who have addressed this, but this is kind of our take on the matter. To address this question, we're going to model this region of the brain with a series of neural networks, and the memories to be stored are these fashion MNIST images, each of which is an example of a concept. Now, we use different methods to kind of convert each of these images into a neural code. So we use a binary autoencoder to turn it into a neural code for entorhinal cortex, and then subsequently some random projection matrices and thresholding to convert them also to binary neural codes from each downstream region in accordance with the biology. And also matching biological observations is that the purple pathway, this mossy fiber pathway, has a higher threshold. So patterns are sparse. And as is known, sparsity is also related to decorrelation. So two closely related images through the purple pathway will have sparser patterns that are more decorrelated compared to the orange perforant pathway. Okay, so this is the neural network setup. And now that we have two encodings for each image, we're going to store them in CA3. And the new thing in this project is that we're going to try to store both of them as a linear combination, unfortunately ignoring some of the nonlinear dendritic integration. But here we just store them linearly with more weight given to the sparse pattern because they arrive more proximally on the dendrites. So that's just storage in this auto-associative Hopfield-like network. And what do we do for retrieval? Well, we are going to queue the network and then crucially run this Hopfield-like retrieval dynamics with either a high or a low threshold. Now, if we use a high threshold, then the network is going to favor recovering the sparse pattern because fewer neurons are active, and conversely, with a low threshold and dense patterns. And then finally, we'll visualize the retrieved memory to see what we get. Okay, so this is what we see for the retrieved memories. When we store more and more examples in our network, through the purple pathway, we see that the network can recover each of them up to a certain limit faithfully, all the different patterns. Now let's consider using a low threshold to recover the dense patterns. We can see that it could recover maybe one that is stored, but if you store more patterns, it gets all blurry and messy. It recovers some trousers and other things. But after storing enough patterns, you can see that the network will recover a sneaker, no matter what sneaker you queue it with. But it's actually the same looking sneaker, and it's very similar to actually this average image formed by just taking the mean of all the sneaker images. So it has built a representation of the concept by accumulating examples. So this is saying that in the same network with the same synaptic weights, just by tuning a threshold, you can recover more of an example type or a concept-like memory. And because it's a relatively simple network, we can use statistical physics to calculate the capacity of our network for each of these types of patterns. And qualitatively, we get similar results, that there is a regime that you can recover both of these sparse examples or the dense concepts. And this is just showing that our theory matches reasonably well with simulation. So the prediction here is that sparsity, whether it's sparse or dense, modulates memory information. So whether it's an example or concept in CA3. So can we test this in any way? To be particular, this is the prediction that our model makes. Each point here represents one neuron. And so our model says that each neuron will convey more information about example identity during sparse phases compared to dense phases. Well, it turns out that in CA3, in the hippocampus, there is a oscillation, the theta oscillation that is prominent. And this oscillation modulates the sparsity of the network. In particular, CA3 is composed of, it contains many of place cells that have place fields. So many CA3 neurons are active in different locations here along a linear track. And so what we predict is that the tuning of these place cells will differ according to whether the network is sparse or dense. In particular, what we do to connect this biological system with our model is we say that we kind of take the leap of faith that the memories that place cells are encoding are positional memories along a linear track here. So the examples of our model we consider to be finer positions along the linear track. So yeah, positions that contain higher spatial information. And just as examples merge into concepts in our model, we consider that these fine positions will merge into a concept of a position, which is maybe a more broad area. So under this kind of assumed equivalence, our prediction becomes about, I guess, position. Our prediction becomes about position information conveyed. And so to extract this position information, what we do is we analyze these place cell data from publicly available data sets. And so this is one place field that we see. And we're looking at all the spikes of this neuron. And we're kind of teasing it apart by different theta phases. And you can see that the theta phase will modulate both the sparsity, the activity of the neuron, and also how much position information it conveys. And indeed, for CA3 place cells, they tend to convey more positional information during sparse phases compared to dense phases. And this is not seen downstream in CA1. So it's kind of a special property of CA3. And this kind of provides some support for our model. So what this model can help to address is this kind of perhaps tension in the idea of what the hippocampus is there for. So classically, the idea of the hippocampus is that it's this, it stamps an index on each unique experience so it can be retrieved separately. But more recently, people have been appreciating that even in the hippocampus, there is some kind of concept building or generalization that is formed. For example, there are these very marked cells, these Jennifer Aniston cells, or here an Oprah Winfrey cell, that will respond to many different modalities of Oprah Winfrey. Presumably, it learns this representation, this generalized representation, by accumulating individual examples. So our model kind of contributes an idea of how the hippocampus can encode information in two different ways. Inhibitory tone can kind of be a dial to twist between the two. Okay, so this was all about the hippocampus. The question that we have from here is that, well, this kind of, I guess, multi-scale encoding is perhaps encoded by the hippocampus. Can this kind of encoding have any performance benefit, or can we even be inspired by it to improve computation in general in certain cases? So to do this, we kind of turn to this workhorse of machine learning, this multi-layer perceptron trained on MNIST digits to kind of classify digits per usual. But we also then augment this MNIST data set with an additional label. This is a set label that is just randomly assigned to each image. So there could be 100 or 10 sets. And so we asked the network to do both digit classification and set identification. To do the former, it has to appreciate the similarities, for example, between this 3 and other 3s. And to perform this kind of set identification task, it has to, I guess, distinguish the differences between this 3 and other 3s. And what we do now is that we can manipulate the encoding in this, this last hidden layer here. We either do nothing, and what this means is that 2 correlated images would probably have correlated encodings as well. We can then apply this regularizer, this decorrelate loss function that seeks to decorrelate each pair of items in a mini batch. And so then these 3s would have very different encodings here. But then in some way similar to what we find in CA3, we then apply this loss function, but only to the latter half of the network, such that these networks would have both correlated and decorrelated encodings. And it turns out that these kind of half-core networks can perform well on both concept learning and example learning. Whereas if you only had one type of encoding, you can be good in one task, kind of at the cost of another. Furthermore, the network can learn to use the appropriate encoding for the more appropriate task. So we adopt this metric of influence by inactivating one of the neurons and then seeing what decrease in accuracy is produced by this inactivation. So we see that for the correlated neurons, they are more influential towards concept learning, and conversely in the other case. And so this is what the network learns to do. It learns to use the appropriate encoding here. So this, in a sense, is a regularizer that seeks to diversify encodings within a single layer. And so I'm rather new to machine learning, so I don't know if this kind of thing has really been done before or not, but it seems like, at least in this very toy example, it can be helpful in cases where you're not really sure what kind of encoding to use. Of course, here we've only used 2 different types of encodings. You can even have a gradient in the loss function weight to have a real spectrum of encoding properties. And this is just for correlation. You can use this for other interesting properties, perhaps like sparsity and other things. So with that, thank you. Thank you mostly to Taro, who is my co-author, and I also will plug a visit to Japan, to Riken. The Center for Brain Science, we have more of like a Berkeley style of campus, but also if you want to visit our AI Institute, this is a view from the bathroom, so it's very much in central Tokyo. So hope to see some of you there. Thank you.
Okay, so we have time for 1 or 2 questions. And Alex, if you can set up, we'll retake your questions. That will be great.
Oh, yes, thank you.
So I have a question. This is not coming just from your talk, but from, I guess, many preceding, theory hippocampus talks. So also like Elafit, I feel often goes in that direction. And my question is basically like, all the findings of play cells and grid cells and hippocampus role navigation mainly come from rodent literature, right?
Yes.
And all the findings about hippocampus role, I wouldn't say just formational memories, but this like very high-dimensional code for experience, like visual code and so on. They all come from primates or humans, right? And I feel there have been like a lot of efforts over the 20 years to try to find, one or the other in the other organism. And as far as I know, they've mostly failed, right? So my question would be basically like, do you think it's possible, right, that finding this overarching theory that's going to combine, like the functional, like the spatial encoding properties and kind of the vision properties of the hippocampus, it's just not a thing that exists, right? They're just 2 different systems that work in similar neural substrate into different kind of organisms, and maybe just trying to find this overarching theory is just not the right direction. I just wonder what you think about that.
I see. Yeah, that is certainly a possibility that perhaps there's a different direction. But I guess for me, I'm more of on the unified perspective, as Bruno says, looking at the conservation of even, the ellipsoid body to head direction cells. And there's been some, findings in rodents that grid cells are tuned to things like sound frequency. And there's some other, place cells that are found, and even grid cells that are found with a lot of data analysis in humans. So I think, just experimentally, often it's easier to show visual and other kinds of stimuli to humans, and then to test animals with place is very convenient.
Yeah.
But hopefully in the future there'll be greater convergence.
So I think we should move on there. So let's thank Luce again.